This slide can get deleted. This one, I mean, these slides are really, again, like pretty awful. So I'm sorry. I apologize for like the really sad animations. But the video that I'm going to show you about the Calvin cycle is supposed to be a little more, I hope this will be a little more helpful than the PowerPoint itself. All right. So you guys are with me, right? You can see this? Yes. Okay. So slideshow, play from current slide. All right. So first we're going to do this. We're going to watch this video. I think that link works. Okay. So you guys can all see this, right?
Nothing replaces a human connection.
Thank you very much. Okay.
You're facing a giant bowl of energy-packed carbon crunchies. 1 spoonful, 2, 3. Soon, you're powered up by the energy surge that comes from your meal. But how did that energy get into your bowl? Energy exists in the form of sugars made by the plant your cereal came from, like wheat or corn. As you can see, carbon is the chemical backbone, and plants get their fix of it in the form of carbon dioxide, CO2, from the air that we all breathe. But how does a plant's energy factory, housed in the stroma of the chloroplast, turn a 1-carbon gas like CO2 into a 6-carbon solid like glucose? If you're thinking photosynthesis, you're right. But photosynthesis is divided into 2 steps. The first, which stores energy from the sun in the form of adenosine triphosphate, or ATP. And the second, the Calvin cycle, that captures carbon and turns it into sugar. This second phase represents one of nature's most sustainable production lines. And so with that, welcome to the world's most minuscule factory. The starting materials? A mix of CO2 molecules from the air and pre-assembled molecules called ribulose bisphosphate, or RUBP, each containing 5 carbons. The initiator? An industrious enzyme named Rubisco that welds 1 carbon atom from a CO2 molecule with the RUBP chain to build an initial 6-carbon sequence. That rapidly splits into 2 shorter chains containing 3 carbons each and called phosphoglycerates, or PGAs for short. Enter ATP and another chemical called nicotinamide adenine dinucleotide phosphate, or just NADPH. ATP, working like a lubricant, delivers energy, while NADPH affixes 1 hydrogen to each of the PGA chains, changing them into molecules called glyceraldehyde 3-phosphates, or G3Ps. Glucose needs 6 carbons to form, made from 2 molecules of G3P, which incidentally have 6 carbons between them. So sugar has just been manufactured, right? Not quite. The Calvin cycle works like a sustainable production line, meaning that those original RUBPs that kicked things off at the start need to be recreated by reusing materials within the cycle now. But each RUBP needs 5 carbons, and manufacturing glucose takes a whole 6. Something doesn't add up. The answer lies in one phenomenal fact. While we've been focusing on this single production line, 5 others have been happening at the same time. With 6 conveyor belts moving in unison, there isn't just 1 carbon that gets soldered to 1 RUBP chain, but 6 carbons soldered to 6 RUBPs. That creates 12 G3P chains instead of just 2, meaning that altogether, 36 carbons exist, the precise number needed to manufacture sugar and rebuild those RUBPs. Of the 12 G3Ps pooled together, 2 are siphoned off to form that energy-rich 6-carbon glucose chain, the one fueling you via your breakfast. Success! But back on the manufacturing line, the byproducts of this sugar production are swiftly assembled to recreate those 6 RUBPs. That requires 30 carbons, the exact number contained by the remaining 10 G3Ps. Now, a molecular mix and match occurs. 2 of the G3Ps are welded together, forming a 6-carbon sequence. By adding a third G3P, a 9-carbon chain is built. The first RUBP, made up of 5 carbons, is cast from this, leaving 4 carbons behind. But there's no wastage here. Those are soldered to a 4th G3P molecule, making a 7-carbon chain. Added to a 5th G3P molecule, a 10-carbon chain is created. Enough now to craft 2 more RUBPs. With 3 full RUBPs recreated from 5 of the 10 G3Ps, simply duplicating this process will renew the 6 RUBP chains needed to restart the cycle again. So the Calvin cycle generates the precise number of elements and processes required to keep this biochemical production line turning endlessly. And it's just one of the hundreds of cycles present in nature. Why so many? Because if biological production processes were linear, they wouldn't be nearly as efficient or successful at using energy to manufacture the materials that nature relies upon, like sugar. Cycles create vital feedback loops that repeatedly reuse and rebuild ingredients, crafting as much as possible out of the planet's available resources. Such as that sugar, built using raw sunlight and carbon, converted in plant factories to become the energy that powers you and keeps the cycles revolving in your own life.
All right, did you guys follow that? Did anyone? Okay, so I'm just gonna go over it ourselves, and then I'll show you this simulation, which kind of helps the whole process. But hopefully, like having seen that, you'll know you'll have some idea of what's coming next. Okay, so basically, the first one is called the light-dependent reactions, right? And then where do the light-dependent reactions happen again? What location? Like
the thylakoid membrane.
Exactly, in that thylakoid membrane, right? And that's where the photosystems, right, are embedded, and that's where the electron transport chain is like passing down the electrons, and that's where the gradient of hydrogen proton ions are is created, and then they diffuse, facilitated diffusion back across into the stroma to create ATP, right? So we did all that yesterday. So the Calvin cycle takes place in the stroma. So that's, again, that kind of like watery space between the thylakoids, right? Between the grana of thylakoids, right? So it's in that like little like watery space in the chloroplast that this is happening, right? And it can be divided into 3 different parts. So you should memorize these 3 different parts. The first part is called fixation. And that's the thing that like duck-looking Dr. Seuss guy was doing, where he was like taking the carbon dioxide and fixing it, meaning like binding it to this other organic molecule already called RUBP, right? So that's the first part, kind of sealing the carbon dioxide into a chemical molecule, right? So getting it from the air and putting it into like a chemical molecule. The second part is reduction. What does reduction mean?
Gaining electrons.
Exactly. So it means that you're adding electrons to that product, right? So that those things are going to be said to be reduced, right? Who's carrying around extra electrons that we remember from the light reactions? Who are those 2 electron carriers that are carrying like extra electrons that they didn't have before in the form of chemical energy? This is where the products of that come in. Who were they?
Was it hydrogen?
Hydrogen, well, kind of hydrogen, but it was like the 2 molecules that we said we were going to use later in the Calvin cycle.
NADPH and ATP.
Exactly. Those are the 2 that are carrying electrons, and they basically deliver the electrons to that molecule that we've created here. So then we say the molecule itself is said to be reduced, and NADPH and ATP basically get oxidized because they're giving away their electrons to this new molecule that we're creating, right? So then first we fix the carbon into it, then we reduce it by adding ATP and NADPH, right? And then we have one of those products that goes out and it can be used to make organic molecules. But the problem is we need to regenerate the stuff that went into the cycle in the beginning, the RUBP, because in order for the cycle to keep going over, around and around, it needs to start with or end with the same stuff that it started with, right? Otherwise, it won't be able to keep doing what it's doing. So that's sort of where that weird math comes in. You don't have to know all the like crazy details about how G3P is then regenerated back into RUBP, but you need to know that RUBP is how many carbons RUBP is, how many carbons G3P is, and to know how many cycles need to be done in order for one molecule of G3P to come out, right? So is anyone, I hope you guys might be totally lost right now, but I'm going to try to show you more like more in like little like animation in a different like, anyway, I'm going to try to show you like with my own animation, like what I'm talking about here. So anyway, the first step is carbon fixation. So what does carbon fixation means? It means that you're taking carbon dioxide from the air and attaching it to a molecule in the plant. Okay, how does the carbon dioxide get in again? Through the
Stomata.
Stomata, right? Good. Okay, so the first thing that happens in this carbon fixation is there's this enzyme called rubisco, right? So this is like the most abundant enzyme, I think, in the world. So it's like, think of all the plant matter and all the stuff, on Earth. This enzyme is like huge in, creating all the oxygen and like the plant life, supporting the plant life on Earth. So very, very important enzyme, rubisco. So this enzyme attaches one carbon from the carbon dioxide. So we take one molecule of carbon dioxide in here, right? And RUBP, which is composed of 5 carbons. So here's RUBP. And RUBP already, like it's in the cell, it's in the plant already. We're not getting it from anywhere. That's just sort of like what the plant starts with. So it has a molecule of RUBP in here, which is 5 carbons, right? Each of these pink balls is carbon, right? And then here's a molecule of carbon dioxide that comes into the air, right? And RUBP, these 5 carbons and carbon dioxide are sort of welded together, like they said in the video, by this enzyme called rubisco, which, so what will the product of this kind of combination be? How many carbons will that product have? If we add a carbon dioxide right in here, it will be what?
6.
6, right? Does that make sense, right? 5 plus 1 is 6, right? So then we have 6 carbon, 1, 2, 3, 4, 5, 6 carbon intermediate, okay? We're good so far? So this is like the fixation stage. And remember, these 5 carbons came from RUBP, which was already there in the plant. And then this one came from the carbon dioxide in the air. And don't worry about where those oxygen go, but those oxygen are reused somewhere else, but that's not the oxygen that's released in photosynthesis. This oxygen gets used somewhere else. The oxygen that's released in photosynthesis comes from the breaking of water in the light-dependent reactions, right? Okay, so now we have this 6 carbon intermediate guy, and actually that guy is pretty unstable. So he doesn't last for very long, or she, if you want to be politically correct. That molecule doesn't last for very long. So what happens in the next phase is that, so this should not say fixation, this should say reduction. I'm sorry. Okay. Okay. So anyway, so we have this 6 carbon intermediate, right? You guys are still with me? Okay. So what happens is that 6 carbon molecule splits immediately into 2 molecules of PGA, right? And it's called 3-PGA, right? How many carbon molecules are in each molecule of 3-PGA?
3.
Good. 3 carbons, right? And we have 2 of them because we originally had 6, so 6 divided by 2 is 3, right? So we have 3 PGA right here. So that just happens automatically, right? And then these 3 PGA, they're not ready yet. So what needs to happen is these guys become reduced. And again, to be reduced, that means what do? They're
Gaining electrons.
taking electrons. And where are they getting those electrons from?
NADPH and ATP.
Exactly. Good. So what happens is NADPH and the ATP from those light reactions before, they reduce 3-PGA, right? Both of these, they reduce those to a new molecule called G3P, which is made of 3 carbons. So it's very similar to 3-PGA, except these, the NADPH and the ATP put their electrons onto 3-PGA to make G3P, 2 molecules of G3P. And then what comes out of that is NADP plus, right? Because now NADPH has been oxidized because it gave away its electrons, right? And ATP has also been, you can say it's been oxidized because it gave away its electrons, and now it's ADP. And what do you think will happen to these 2 things again? Where will they go?
Back to like the light dependent reaction.
Exactly. They're going to go back into the light dependent reaction so they can keep like regenerating this whole thing, right? So those, again, that's where these guys are used. They're used in the reduction phase of the Calvin cycle, right? So 3-PGA is reduced to make G3Ps, 2 molecules of G3P, which are 3 carbons each, and then they are recycled back to the light dependent reactions, right? Okay. We're good so far? Yes? You're following where the carbons are, and obviously it's not as simplistic as this. They have like phosphates attached to them and all this other stuff, but I just want you to kind of keep track of the carbons and where they're going, right? So you guys you understand that so 5 of these carbons came from RUBP, 1 of them came from the carbon dioxide molecule that was added, right? Okay, so then we have these 2 molecules of G3P, and 1 molecule of G3P goes out to then go make organic compounds, right? So G3P is like a precursor to like more like higher level organic compounds like glucose, right? But the problem is what we need in order for this cycle to always be going, what do we need to do with this? We need to go regenerate. What do we need to go regenerate? What did this cycle start with again?
Carbon dioxide.
Carbon dioxide is just like coming from the air. So we don't need to regenerate the carbon dioxide. But in order for the carbon dioxide from the air to be added to make this guy, what did we need to add to that?
Oh, NADPH and ATP. Oxygen.
So here's the carbon dioxide that we got, right? That we put in. But what was this thing? Where did all those other 5 carbons come from?
Oh, the RUBP.
Yes, that's exactly what it was. So we put in 5 atoms of carbon in the form of RUBP. So we need to get back to that in order for this cycle to go again. So Rubisco can bind 5 and add 1, right, to make that 6 carbon intermediate, right? So, but that's a problem because if we take 3 out, right, to make a G3P to go make an organic molecule, how many do we have left to go back into the cycle? If we take 1 G3P molecule out, so this is what, so basically what happens, is we have to take 1 G3P out to go make organic compounds like glucose or whatever, other carbohydrates, right? But then we need to regenerate the RUBP, but is 3 enough to generate RUBP?
No.
No, how many do we need?
5.
We need 5 total, right? And so that means we need 2 more. Where are we gonna get those 2 more? So just think about what you're putting into the cycle. What did we add to the cycle? How many carbon dioxides do we add to the cycle in the beginning?
1.
Just 1. And then, but if we want 3 of these things to come out, how many carbon dioxides do we need to add to get a 3-thing product that we can just go and take out?
You mean 5? Wait, do you mean 5?
So, okay, so it starts with 5 in it, right? And I want to get a net gain of 3 that can come out of the cycle without stopping the cycle, right? So I put 1 in already, and then I want to get 5. I still want to get 5 like for each 1 carbon that I put in, right? But I need to get a 3 carbon product that I can just take out and use elsewhere. So how many carbon dioxides do I need to add in order for me to take out 3? So think about that.
2.
Huh?
Is it 2?
Close.
3?
3, exactly. Because think about it. You're like, the other 5 are just cycling around. They're being they're being converted, but then they're being reconverted. But you need a net. If you're going to take a 3 carbon molecule out of here, you need to have added 3 carbon dioxide, right? So in order to get 1 molecule of G3P that you can then take out of the cycle and not recycle back into making RUBP, that means you need to have ran the cycle 3 times, right? Meaning adding 1 carbon dioxide molecule each time, right? So how many carbons do we put into the cycle? Originally, we put in 1, right? But in order to get 3 out, you have to run it 3 times. to create one G3P molecule, right? Suppose I want to make a molecule of glucose. How many carbons is glucose? How many carbons is glucose? What's the formula for glucose?
Isn't there 6 carbons?
Yes. So how many? So that means I need how many G3Ps?
2.
2 G3Ps, right? Which means I need a total of 6 carbons to be added. So how many times do I have to run the cycle to get one glucose molecule?
6 times.
6, exactly. So to get one G3P, I need to run the cycle 3 times, right? Because think about it, like the thing that you're taking out, you have to have put in, right? So if you want to take out a 3-carbon molecule, then you need to have added 3 carbons, not counting the RUBP, because RUBP needs to just be recycled as is, right? So you need to have added 3 carbon dioxide. If you want to get a glucose molecule, which is C6H12O6, right? 6 carbon molecules or atoms, then you need to have added 6. So the cycle needs to go 6 times for every glucose molecule that we're adding, right? So if we take one G3P out to go make organic compounds, that means we needed to have run the cycle 3 times for every G3P molecule that we get. And if we wanted to take out 2 G3P molecules to make a glucose molecule, then we needed to have run the cycle 6 times. Okay? Are you guys sort of with me or totally lost? Okay? Yes? No? Okay, that's pretty much it. Oh, there's one more slide. And this is just like an overview, which I hope you guys can sort of understand a little better now. So just look at this picture real quick, all right? So here we're starting in the beginning. So we're adding, they just went ahead and said, okay, we're gonna add the 3 CO2s right here. They didn't make you even think about it. They just wanted you, they're just telling you, we're adding 3 CO2s, right? So how many different RUBP molecules are going in? Because RUBP is 5 and each CO2 only gets added to an RUBP, right? So it's not like you add the 3 CO2s together. It's like you add one CO2 to one molecule of RUBP, then you add another CO2 to another molecule of RUBP, and you add another one to another molecule of RUBP, right? So it's like, so then you get 3 of the 6 carbon unstable molecule that then divides into the 3 PGA, right? The 3 PGA then gets reduced by adding ATP and by adding the NADPH, right? So those 3 PGA ones are reduced to G3P. And we have 6 of them now, right? And then 5 can go back and regenerate 3 RUBP molecules. And then that one G3P can come out as an organic molecule and then go contribute to being glucose or something like that, right? I want you guys to just go back and look at this and think about it because it does make sense if you really think about the numbers. You might want to take like little cotton balls or little marbles or something like that to kind of try to get you to understand a little better too, because obviously you can't create or destroy things, right? But if you want the cycle to keep going, you need enough of those little carbon molecules to then reform the thing that started the cycle to begin with. So you need this RUBP to complete the cycle, to start the cycle and complete the cycle, right? So make sure that you don't, if you only add one carbon dioxide to one RUBP molecule, you can't get a G3P out because you wouldn't be able to regenerate the RUBP. Okay? All right. So where does this Calvin cycle happen? Can someone remind me where the Calvin cycle happens?
Stroma.
In the stroma. Very good. Okay. And what goes into it? So what are we putting into it? So that is sort of being changed, right? RUBP doesn't go in or come out because it's just being recycled. What about Rubisco? What type of molecule is Rubisco again? Do you guys remember?
An enzyme.
Enzyme. And what's the rule about enzymes when they do their work?
They could be reused.
Exactly. They don't get used up. So they can be reused. So we're not really counting Rubisco as going in or out, and we're not counting RUBP as going in or out because they just are, going around, right? But what is going in is carbon dioxide, right, which is being let in through the stroma, I'm sorry. This is in the stroma. It's being let in through the stomata, and then also ATP and NADPH from the light reactions are coming in to reduce that, right? And what comes out of the Calvin cycle then? What are we getting out of the Calvin cycle?
Glucose?
Not exactly. So the glucose doesn't come out of the Calvin cycle directly, but G3P, which is the precursor to glucose, comes out of the Calvin cycle, right? So it's really G3P. And also what else is generated? ADP again comes out, and so does NADP plus. Okay. What enzyme is used to fix carbon dioxide in the fixation stage?
Rubisco.
Rubisco, good. And then, yeah, what is the product of the Calvin cycle? So it's G3P, which is the precursor to glucose and other organic molecules and like other carbohydrates and stuff like that. Okay. All right, so I want to show you guys like one last thing, and then I'll let you go and study. What am I doing here? Okay, so this thing, okay? So here's like a little, everyone can see that, right? Let me see if I can, work. Okay, so you see this, right? So this is kind of like a simulation which shows you what is happening. So I'm going to click stage 1. So stage 1 is fixation, right? Do you see here's rubisco, here's carbon dioxide, and here's the ribulose biphosphate, which is RUBP, which is how many carbons again? 1, 2, 3, 4, 5. 5, right? And do you see they have these phosphates on the side? Biphosphate, bi means 2, right? And so it has 2 phosphates. I've seen it biphosphate, I've seen it bisphosphate. You know, you don't need to, I'm not going to hold you accountable for that. But you need to know RUBP, right? And that it's 5 carbons, and that rubisco will combine RUBP with carbon dioxide from the air, right, to create a 6-carbon intermediate that immediately breaks down into 3-PGA, right? 3-PGA for each carbon dioxide molecule. So if, we have 2-PGA for every carbon dioxide molecule or every RUBP. But if you put 3 carbon dioxide molecules and 3 RUBPs in, then you'll get 6 3-PGAs, right? All right, so let's just watch this and see what happens if it lets me do this. Okay, so there's the fixation. Do you see how there's one 3-PGA, there's another 3-PGA, there's another 3-PGA, right? Is everyone okay with that? Everyone's comfortable with that? Okay, and now they want me to continue. So the next is the reduction. So NADPH and ATP convert 3-PGA, which are all these, to G3P. So let's see what that looks like. So there's the ATP and the NADPH working their magic on this and then eventually reducing it to G3P, right? ATP, G3P, NADPH. Okay, you see. So now we have 6 G3Ps, right? But how many do we need? We want to take one of them out of the cycle, right? So this one is going to go off and make sugar by itself, right? So we have to say goodbye to this one. Does it work out though? Because we added 3 carbons in these 3 carbon dioxide molecules, right? And then the 3 carbons are now being taken out of the cycle, right? But the rest, right? So how many carbons are there total in here within the rest of the G3Ps? There are 5 more G3Ps left. So 5 times 3 is
15.
15, right? And then we have 3 of these, but each of them are 5. So again, total of how many carbons here?
15.
15, right? So you see how this all gets recycled back into this, and all these 3 that were added in then are kind of coming out in this form over here. Okay, but in order to get this, we have to make all 6 of them and then reconvert them back into this. You can't just say, okay, I'm going to convert these 3 carbon dioxides directly into these this one G3P molecule. Doesn't work that way. They have to kind of go through this whole process of creating all of them and then just taking one out and then generating all of them back to the RUBP form, right? All right. This quiz is like, it might be really hard. So if you don't understand this, I think you should definitely talk to me about it. I can stay after, class today and go over some stuff because we still have a little more time. If you understand it and you feel like you're going to do well on it, that's fine. You can go and, oh my gosh, Bucky's a little indecent there. Okay. Yeah, so, yeah, right? Does everyone feel okay? Please, if you don't understand this, we can go over the light reactions and the Calvin cycle one more time. I can give you an extra worksheet for you to work on to kind of like solidify these concepts. But if not, then you guys are free to go and we'll do the quiz tomorrow, right? So the quiz tomorrow is on, now be careful, it's on, what was our quiz on last time? We didn't do a quiz last time, did we? Did we do a quiz last week? Oh my gosh, I'm so confused. Let me see. We did, yeah, when was our last quiz? Did we do a quiz? Can someone?
I think we did one last week.
We did, right? And that was on plants, or no, that was on, no, we did one on Thursday on the fungi and protists, right? Was that right or no? Yeah, I think we did.
Yeah.
Yeah, fungi and protists. I published that. I released those scores, right, to you guys? Because I'm getting all disoriented. Okay, so yeah, tomorrow, I mean, I'm warning you, this is going to be rough for some of you. So make sure you really know where each part is located, right? Where you have to know, what the sequence of events is in photosynthesis, which photosystem is hit first, which side of the thylakoid membrane does the gradient, form on, right? What type of transport are the protons being like pushed from the stroma to the lumen? What kind of transport is that? When the protons go from the lumen to the stroma, what kind of transport is that, right? So you need to know, all these like sort of pretty specific things, and you need to know the plant stuff too. So plants, energy, photosynthesis. This is kind of a biggie. So make sure you know it, right? Do you guys feel confident about it, or do you have to study a little bit? It's a rainy day anyway. You don't need to go outside. You can just study. And you don't need to watch Outer Banks either. Outer Banks isn't that good. No, I'm just kidding. Anyone? No? Okay. Is anyone staying with me to do an extra worksheet right now? No? No takers, really? Okay. All right. Well, or no one wants to admit it. Okay. Well then, good luck. Have a good afternoon, and I'll see you tomorrow for the quiz. Okay?
All right, thank you.
Bye, thank you.
Bye. Thank you.
Bye.